Today, we will discuss multicellularity versus supracellularity. Multicellularity is a highly diversified and widespread phenomenon that cuts across all the domains of life, spanning from bacterial biofilms to plants and metazoans. Traditionally, multicellularity was mainly attributed to complex organisms. More recently, it has also been used for simple microbial colonies or biofilms. Red algae is the first known appearance of eukaryotic multicellularity. In some cases, the group of cells is no more than a collection of individual cells, but in higher plants, the group behaves as a supracellular unit, whereby the cell group could be considered as a joint supracell, the concept of which was conceived over a century ago. The development of recent tool has also provided considerable evidence that cell collectives are highly cooperative. In supracellular arrangements, cells are integrated with marked front to back specialization and continuity of cytoskeletal structures between neighboring cells, ensuring mechanical coupling. Such collective polarities implies communication between cells to coordinate subcellular processes. First, we'll start with the multicellularity. Multicellular organisms composed of many cells, which are of varying degrees integrated and independent. The development of multicellular organism is accompanied by cellular specialization and division of labor. Cells become efficient in one process and are dependent upon other cells for necessities of life. So specialization in single-celled organisms exists at the subcellular level. That is, the basic functions that are divided among the cells, tissues, and organs of the multicellular organism are collected within one cell. Multicellular organisms are composed of more than one cell, with the group of cells differentiating to take on specialized functions. For example, in humans, cells differentiate early in development to become nerve cells, skin cells, muscle cells, blood cells, and other type of cells. One can easily observe the difference in these cells under microscope. Their structure is related to their function, meaning each type of cell takes on a particular form in order to best serve its purpose. When the entire organism emerges from a single cell due to the cell division process, all cells of the resulting organisms are clones. So this type of multicellularity is referred to as clonal multicellularity. We all are clear examples of clonal multicellularity, as well as plants, fungi, and all groups of algae. Bacterial domain also includes some groups of clonal multicellular organisms, the most prominent of which are filamentous cyanobacteria. Their filaments include tons of cells which are tightly interconnected and exchange their metabolites by microplasmodesms, analogous to plant plasmodesms. Another case of multicellularity is aggregative multicellularity. The best example of it is Dictyostelium dicotyledonum. In a stated state, it is just an amoeba. But upon starvation, hundreds of amoebas come in flock in one point and aggregate to form a multicellular organism. It further transforms itself into a floating body, which in turn sporulate and gives rise to a new generation of amoebas. The essential trait of aggregative multicellularity is developing a whole organism by aggregation of genetically heterogeneous individual cells. Now, question arises, how can a multicellular unit arise from single cells? The answer lies in the way each cell communicates with the other via cell signaling. For multicellular development, cell signaling is crucial and can occur in various forms. For example, signaling molecules may be secreted from the cell to reach target cell receptors embedded within the lipid membrane and exposed externally for other cell receptors to bind and respond. Through these signaling molecules, cells can communicate with each other and allow for complex patterns to function. Cell signaling is crucial not only for multicellular organisms to function as a unit, but also for unicellular communication. Now listen, in multicellular organisms, all cells begin life as stem cells. Stem cells? Stem cells are the cells in the body that can develop into many different types of cells. Why these cells are stem cells? Because they are self-renewable and unspecialized. So, in multicellular organisms, all cells begin life as stem cells, which then either remain stem cells to produce more stem cells, or they go through the process of differentiation, whereby stem cells becomes a specialized cell. That is a cell taking on a role to have specific functions. This is why a neuronal cell is different phenotypically and functionally to a skin cell. This allows multicellular organisms to be far more complex than unicellular organisms, as they can compartmentalize their function to different cell types, enabling larger and more complex uh, life forms to exist. Now, as I already told you, for multicellular development, cell signaling is crucial. So, in the case of higher plants, plasmodesmata connect adjacent cells and allow communication to regulate differentiation and development. You can see here this first diagram, plasmodesmata. This is plasmodesmata. In the case of animals, two main general structures that enable short and long range intercellular communication are gap junctions and tunneling nanotubes. In the case of fungi, filamentous fungi have also developed specialized structures called septal pores that allow intercellular communication via cytoplasmic flow. So these are two different types of septal pores in the case of filamentous fungi. Now we will discuss supracellularity. Like other complex multicellular organisms, plants have cells with distinct identities and functions, and these are organized into tissues and organs. Different plant organs are exposed to vastly different environments, depending on whether they are aerial or subterranean, underlining the critical need for communication to mediate coordination within plant. In 1897, Wilhelm Ferrer noted that for a plant to coordinate its physiological responses across the whole, there must be continuity throughout the entire organism, and that connections between cells must transport material and messages between tissues. Now, supracellularity. I will explain this phenomena with the help of a diagram. Just look at this diagram. In animals, now life uh, of a plant and animal start from a single cell. In animals, cell cleavage is complete, right? Means why plants are supracellular. Just listen. In the case of animal, cell cleavage is complete, and newly formed cells are both motile and in control of their fate. In plants, cells are non-motile and maintain their cytoplasmic continuity with neighboring cells with the help of plasmodesmata. Their fate is controlled at the level above that of the individual cells. Right? This, whereas animal development, it is clearly multicellular in nature. But plant form and function is orchestrated at the supracellular level, right? Animals and plants both seem to need to maintain a degree of simple cytoplasmic continuity. In animal, this function is fulfilled by gap junctions, while in plants, plasmodesmata fill this role, right? So here, in the case of plants, all cells are interconnected with the help of plasmodesmata, right? Furthermore, why plants are supracellular? Plant cells are surrounded by cell wall. Here, plasmodesmata create an interconnected multicellular cytoplasm that traverses cell wall and almost all tissues of the plant and is referred to as symplast. Right? Now, symplast uh, is the interconnected cytoplasm of plant cell, enclosed by cell membrane and plasmodesmata. It is considered the living part of the plant tissue without cell wall or intercellular spaces. Symplast involved in many processes, including transporting nanoparticles, then communication and signaling, water flow, reproduction, and RNA interference. So, plasmodesmata connect the symplast directly to the phloem, which serve as long distance conduit within the vasculature. Just look at this diagram. This concept of supracellularity will be more clear to you. Now, just look at this diagram. Here, plasmodesmata establish interconnected cytoplasm called symplast, right? Through which metabolites, proteins, and RNAs are exchanged between cells, tissues, and organs. So, soluble molecules can move from cell to cell in which they are synthesized into the surrounding cells via these plasmodesmata. Plasmodesmata are the plasma membrane lined tubes that connect neighboring cells across the cell wall. This is plasmodesmata, right? Now, look at this diagram. Plasmodesmata can open or close via deposition of callus, this blue in color, in the cell wall that surround the pore, right? This is regulated in response to a variety of developmental and environmental signals and control the passive movement of the molecules through the plasmodesmata. Now, here specialized plasmodesmata called pore plasmodesmata that connect companion cells, CC, companion cells with sieve elements, right, in the phloem. Now, pore plasmodesmata have asymmetric pores. They have more opening on the companion cell phase and means than into the sieve elements phase, and this facilitate high volume translocation of sugar into sieve elements. In sink tissues, here the phloem unloads via phloem pole pericycle cells, and here the plasmodesmata at the interface between sieve elements and the phloem pole pericycle cells here are funnel shaped with the wide opening on the sieve element face. So these are the different type of plasmodesmata and how this plasmodesmata connect different cells in the case of plant. So here plasmodesmata create an interconnected multicellular cytoplasm. All means that traverse cell wall also and almost all tissues of plant and is referred to as symplast, right? So plasmodesmata connect symplast directly to the phloem, which serve as long distance conduit within the vasculature. So that's why plants are supracellular in nature. Thanks for watching. If you like this video, make sure to subscribe for more. Thank you.